Yeah, sure.
So hello, everyone. Thank you for joining today. So this new seminar today in CIS seminars, we have Manuel Rausch from University of Texas at Austin. Manuel is originally from Germany, but he has developed all his research career, I would say, or yes, 100% research career in the US. So he graduated his PhD from Stanford in 2013. He spent 2 years in industry in research and development of medical devices. And then in 2015, he joined the lab of Jay Humphrey as a postdoc during 2 years. So Jay Humphrey, for those that are not on the biomechanics side, is very well known in biomechanics, especially soft tissue biomechanics, and is actually a collaborator of Stefan Avril and other colleagues here also. And since 2017, he's an assistant professor at the University of Texas at Austin. He also has an appointment at the Ogden Institute of Computational Engineering. And he's developing, well, he has his own lab, and he continues his work on soft tissue biomechanics, both experimentally and computationally. And he has been testing and reporting methods for many types of tissues, but lately he's been interested also in clot mechanics, which I think it's a very interesting topic. And I think this is the topic of his talk today. So thank you very much, Manuel, for joining us. I'm especially grateful because there is a big time difference also with Texas. So thank you for that. And also considering that they've been having snowstorms the last weeks, and that there's been logistics problems, I believe, related to all that. So thank you very much, Manuel, for being here.
I very much appreciate the invite and also the kind words. You know more about my career than I do. I don't know where you found that information, but anyways, I appreciate it. Yeah, so as said, I recently started studying blood clot mechanics. Well, recently, I started that during my postdoc, but kind of gave up on it and started, when I started my appointment here, doing sort of things that I was more familiar with. And as I was kind of gaining a footing and became a little more confident, I started this new project. And so this is work that I've collected, or data that I've collected over the last year with the help of a number of collaborators. Some of them are my students, some of them are colleagues, and I'm going to introduce them accordingly. But first, I wanted to just give you a quick overview what sort of my research philosophy is and what I do in my lab. Overall, we're interested in right cardiovascular disease, and I'm going to speak a little bit about what that means. And we do kind of a little bit of everything, nothing really well, but all decently okay. We do nonlinear imaging, such as 2-photon microscopy. We do numerics. We do a lot of experiments recently, and we do animal models, small animals, rodents, mice, and rats, as well as large animals and sheep. And while we're interested in using sort of the methods and to inform basic science, ultimately our goal is to develop predictive models to improve disease prognosis, diagnosis, as well as therapy. And so when I say right cardiovascular disease, if you're not too familiar with the cardiovascular system, then I'm just going to give you a quick overview. You sort of have your heart, and it has 2 chambers, well, 4 chambers technically, but generally speaking, 2 working chambers. One is the left heart, sort of on the left side from your own perspective, and one is the right heart. And so the left side of the heart is what I refer to, that comprises the left ventricle, the mitral valve, the aortic valve, and your arteries. On the right side, we have the right ventricle, tricuspid valve, pulmonary valve, and sort of the venous vasculature. So I'm interested in diseases of this right side. And this is not the reason, but this is sort of a good reminder of why it's important to study the right side. During this COVID-19 pandemic, you may have followed this a little bit, but there has been a huge increase in incidences of thrombotic complications, diseases. And interestingly, also, the disease seems to be affecting the right ventricle much more so than the left ventricle. To be quite honest, I haven't stayed up to date with all the literature. I mean, it's incredible how much data and how much research there's been going on in just the past couple of months. But as far as I understand, we don't fully understand how the right ventricle is affected and why. And so this just reminds us that we need to know more about these organ systems, because they've historically been kind of ignored. And so in my lab, I follow sort of 3 research directions. I do right heart mechanics, tricuspid valve mechanics, and then blood clot mechanics. And today I'll be talking about this project on blood clot mechanics. In this work on blood clot mechanics, I'm motivated by a specific disease. And so I guess the overall question is, why would you study clot mechanics? And the disease I'm interested in and that I think needs to be addressed is that of pulmonary embolism. And I'm going to talk you through this a little bit here in just a second. It's pretty superficial, so if you have more questions, please let me know. I'm super happy to address them. And also on that note, I don't mind being interrupted at all. In fact, that gives me an excuse to lose my thread and then blame you for it. But in all seriousness, I don't mind at all if you ask and interrupt. So blood clot mechanics, why study it? Well, blood clot mechanics actually plays critical roles in physiology and pathology. You may know this, but the fundamental function of blood clot is when you cut yourself and you bleed through a severed vessel, obviously at some point you hope that your blood stops flowing. And the reason it does stop flowing are multifold, but one of them is that your blood locally senses that there's damage to the vasculature, and it actually activates something called the coagulation cascade, which transforms your blood into this clot. And the clot basically occludes this hole in your vasculature. So obviously, its mechanical function is important here because it has to be flexible. It has to accommodate sort of the softness of the surrounding tissue, but it also has to withstand the hemodynamic forces, the pressures, as well as sort of your locomotion, right? You move around, you don't want the blood clot to break off and you start bleeding again. In disease, it also plays a very important role, and specifically in a disease called pulmonary embolism. And here on the right-hand side, you see sort of a artistic depiction of this disease. You have here a vein in blue, and for one reason or another, a blood clot may form in your veins. And here they actually depict a venous valve. You may not know, but your venous vasculature actually has valves that prevent sort of backflow, because it turns out that your venous vasculature operates under very low pressures. So if you stand upright, the hydrostatic pressures are higher than the gradient that your venous vasculature actually has to move blood. And so what it does is it actually uses your muscles that contract and creates a peristaltic pump to move blood upwards. And then to prevent it from basically sinking back, you have these valves. But it turns out these valves are hemodynamically kind of complicated, and you have recirculation areas behind them, which lead to hypoxia and then biochemical processes in these cells lining these vessels that ultimately lead to this thrombus formation. Long story short, the thrombus sits here and it can break off. And when it does break off, it sort of travels downstream and eventually can end up in your cerebral vasculature and cause a stroke, or in your in your coronary vasculature and cause a heart attack, or in your lungs and cause a pulmonary embolism. And so ultimately, this embolization event is about breaking, right? And so mechanically speaking, we refer to it as a fracture problem. And if you've ever studied fracture, you need to know a lot about a material to be able to understand and predict its fracture, especially a material that's as complicated as thrombus. And we'll be talking about why it is complicated. So with all this being said, you may not really have a good appreciation for what thrombus looks like as a material. And so here is kind of a depiction of this. So this is a scanning electron microscope image, and this is about 10 micrometer, a 10 micrometer scale bar. And what you see here is sort of this fibrous web. And this web is actually a semi-flexible polymer called fibrin. And this semi-flexible polymer actually forms during this coagulation. So the monomer is called fibrinogen. It just flows around your blood all the time. And when the coagulation cascade gets activated, it actually triggers the polymerization of that monomer into this web. What you also see are these blobs here, and they're essentially red blood cells. Well, not essentially, they are red blood cells, but they essentially act like passive fillers in this sort of hydrated composite material. And then there's also active fillers. They're called platelets, and these are pseudo cells. They're not really cells because they don't have nuclei, but they have a membrane and they can actually get activated. They have a contractile apparatus that kind of tensions the whole web. And then, of course, a lot of water. So it's a pretty complicated composite material. So when I speak about blood clot mechanics, there's of course shoulders of giants that we stand on. So to be clear, these are sort of the shoulders that I'm referring to. So this is work that's been done on whole blood clot, and this is ignoring almost 100 years of work on fibrin hydrogels. The reason we don't use fibrin hydrogels to better understand and study blood clot is because it turns out that they're actually quite different. It makes a huge difference whether you add red blood cells and platelets to fibrin or you study fibrin by itself. But if there's all this great work, why are we not just happy with that and we analyze those data? Well, it turns out that blood clot mechanics are really complicated, and they depend on the species. They depend on which vessel the blood clot was formed in. They depend on the location where it was taken from. It depends on the disease under which it was formed. It depends on how long it's been there. And it depends on how it was tested. And so this is actually pretty frustrating because I can go to all these papers and I find a lot of confusing and contradictory information. Some people find that blood clot is strain stiffening. Some people find it's not. Some people find it undergoes a negative point-getting effect. Other people find a positive one. And so it's entirely unclear, sort of as a comprehensive set of data, what is going on. And so we essentially decided to use blood clot, in vitro blood clot, to kind of try to get a comprehensive, sort of all-encompassing perspective on blood clot mechanics. So when I first decided to take this a little more seriously, I kind of, in my mind, created this task list of things that I would want to look at. And so this is kind of what I came up with, right? I said, okay, let's make some clot in the lab, and then let's look at its sort of hyperelastic behavior. And, you know, meaning we approximate it as a hyperelastic material. We ignore sort of all the complicated viscoelastic stuff and just kind of look at how it behaves. And then the next step should probably be to sort of look at its viscoelasticity. And then ultimately, if we want to model a fracture, we should probably look at damage also as a dissipative mechanism. And then ultimately, of course, we want to look at fracture. And the reality is, if we truly want to understand this material, we should not only look at the macro scale, but the micro scale. And so over the last year, I think we made pretty decent progress on sort of these areas and not so much on the other areas. And that's actually, you know, my ambition for this and the coming years. And so this is what I'm going to talk about. I have two graduate students that have been outstanding. Ella, she's the experimentalist. She does a lot of the experiments. So Therese is a computational guy, so he helps them with the computational work. And then Sapin and Birkin are colleagues here at UT. Sapin is an imaging guy. Birkin is an numerics guy. And I've actually talked them into blood clots. And then Adrian is a friend and colleague of mine who also recently started working with me. So he has a student, John, who helps out here as well. So I want to be very clear that in this work, I'm supported by a great team of friends and collaborators and students. So as a first step, I figured what we'll do is we'll first try to figure out how to test these blood clots. Because blood clot, I'm not sure you have an intuitive sense for it, but it's kind of like Jello, right? It's very soft in the sense that it has very low stiffness. In contrast to Jello, though, it's actually not very brittle. So Jello actually behaves pretty brittly. Blood clot does not. It's a pretty ductile material, but it's incredibly hard to handle. And basically, the moment you handle it, it loses its water and it changes its mechanics significantly. So the way we came up with testing it was basically we 3D print these casts, and we actually line them with Velcro, the sort of like, you know, material that you use to close your tennis shoes when you're, you know, 5 years, 6 years old. And so basically you have these little hooks that are being submerged in this blood clot. And then we let the blood clot coagulate, meaning solidify. We do this in an incubator at body temperature. And then we can remove that. And then we have this kind of blood clot. And here you see the photograph. So this is 10 millimeters, 10 millimeter cube. And it turns out that we can put this in a really nice shape. And so that kind of makes it a neat material to work with. We can cast it into any shape. But now we have this Velcro on top and bottom. We're not showing this here on the right. And then we can glue this Velcro to sort of holders. So the first test we actually did was a simple shear experiment where we glue this to pin stubs and then we just shear the material. And so what we see here on the top left is a representative data set. And this is actually a truly representative data set. It's not, you know, the best one we could pick from our set. But what you see is this very classic strain stiffening behavior that we see in a lot of soft materials. And actually, the reason most likely being that that is driven by the semi-flexible polymer network. We show here actually an upstroke and downstroke, right? So in gray, we see the hysteresis, and it's actually quite minimal. And I'll talk about this in just a second. And so to analyze this as a first approximation, we sort of take the midline of this, right? We ignore that there's hysteresis as a viscoelastic effect, and we just assume that this behaves like a perfectly elastic material. And so the side note is that I actually chose simple shear as the first way to approximate this, because simple shear is approximately volume preserving. And so we hypothesize, and I'll talk about this more in the next couple of slides, that a lot of the viscoelastic effects are due to sort of the poroelasticity of the material. So if the material is volume preserving, then sort of the internal pressure gradients are probably minimal, or at least smaller than under other deformation modes, and therefore the viscoelastic effects should be minimal in these experiments. One way that we characterize these materials often is, well, they're nonlinear, so there isn't really a Young's modulus we can use to characterize the material. So we often sort of approximate two different stiffnesses, one at small strains, which we call the toe stiffness, and one at larger strains that we call the calf stiffness. So if you hear me talk about this, this is what I'm referring to. And then we actually loaded this under a triaxial load cell, which is neat because then we could also measure the normal force. And so we here actually see that we produce a force that tries to pull these pin stubs together. So it produces a negative pointing effect, which turns out is quite significant because that's different from fibrin meshes. And presumably, and others have discussed that, that is due to the existence of these fillers, the red blood cells and the platelets. One trouble you have when you work with biological materials is that they're biological. So blood doesn't stay good forever. And we were actually interested in sort of seeing to what extent we can test blood over at least 3 days. And we counted red blood cells as well as platelets and made sure that they don't die at a rate that's significantly faster than over the period over which we tested. And we do find that they actually do die a little bit, which is not great. But it turns out that the toe stiffness and the calf stiffness, they're actually approximately constant over those 3 days. But you also see that the error margins are pretty large. But regardless, you see that at small strains, the stiffness is in the Pascal range, which is incredibly low. And at larger strains, it's in the kilopascal range, which still is very low, but obviously stiffer, reflecting that nonlinear behavior. And so ultimately, we recognize that to understand the fracture of a complex, hydrogel under large deformation requires numerical models, right? There's almost no other way to understand, say, quantities like fracture toughness without having models that allow us to decouple other dissipative mechanisms from that of surface creation. So our goal was to identify, first of all, sort of a hyperelastic material that can act as our backbone to this sort of modeling pipeline. And we compared sort of classic examples. We looked at the Octa model, the Fung model, as well as the Yeo model. And so these are all hyperelastic models. The Fung model has an exponential function. The Yeo model is a polynomial function, and so is the Octa model. If you have questions about these specific model formulations, I'll be happy to talk about it. But it turns out that they all fit our data pretty well. And so this is just one representative sample. We actually did this on 20 or 30 or so. You can find the raw data in this publication down here. But what I found was interesting was that when we fit the normal functions, right? And so these are simultaneously fit, obviously, not individually. It turns out that the Octa model just did a much better job in capturing the normal stress as opposed to the Fung model or the Yeo model. And so moving forward, we'll be using the Octa model. And in the paper, I discussed this a little bit, but we in the biomechanics field have also just sort of by default gone to this sort of Fung type, Holzapfel type model. And it turns out that that's not always necessarily best. And that was just an interesting finding. So moving forward, we'll be using the Octa model. So now that we kind of have a very fundamental understanding of blood clots, we know it's strain stiffening, we know it undergoes a negative pointing effect. We've identified a hyperelastic material model to capture sort of the elastic effects. We wanted to start looking at a mode of deformation under which we could study fracture. And so if you look at classic fracture experiments, sort of a mode 1 experiment, you see these geometries that people refer to as pure shear experiments, right? And so our goal was to say, okay, and we should start looking at this deformation mode if that's what we ultimately want to use to characterize fracture. And I want to reiterate, the reason we're interested in fracture is because we're interested in this disease called pulmonary embolism, where embolization really just means fracturing. And And so similarly, we can basically cast blood clot into this shape. And this is again Velcro, and we can put this in this custom sort of holder. And then that holder can be attached to a uniaxial tensile testing machine. And so we can just do your classic uniaxial experiment. So I have to sneeze for just 1 second. Or not, apparently. Okay, sorry about that. And so what we said here was, okay, we're going to look at the viscoelastic approximation of this material next. And if you sort of think about your classic viscoelastic effects, you think about strain rate dependence, you think about hysteresis, you think about stress relaxation. And so these are the modes under which we wanted to look at this. So first, we just loaded the thrombus to, a pretty large strain. So I call it effective strain, which is basically the clamp to clamp strain. And we do this over, I guess, 2.5 decades, right? So we started 0.2 millimeters per second, and we go all the way up to 5 millimeters per second, where this was really the highest rate that our uniaxial tensile testing machine can do. And we see a moderate effect, and we can actually, so these are about 6 samples. The error bars are very small, and we see a very small statistical effect between them. But, overall, I wouldn't say that over these ranges that kind of reflect the deformation rates in vivo, that we see a huge difference. Next, we underwent the cyclic loading. And of course, here you have sort of your native loading curve, and then you have your unloading, and then you have your reloading, right? And so one thing, of course, you find is that you have some sort of stress softening, right? You don't return on the same curve. You have also hysteresis. And you have this kind of interesting effect where when you return to your 0 point, you kind of have negative stresses here. And so in the second experiment, what we did was we instead stopped at 0 Newton rather than at 0 millimeters. And then you can kind of quantify what I call permanent set here, which is undetermined whether that's permanent or temporary. And that's something that we're about to look at. So is this a result of damage, or is this just a viscoelastic effect? We don't know at this point. But of course, you also see sort of the strain rate dependence in these data. Something else we're interested in was to see how does this material actually change over repeated loading. So we loaded this over 10 cycles, and we looked at things like the hysteresis work, right? So basically the area under the curve that's set, how does this change, as well as the peak force. And we see that there's sort of an initial change, and then it kind of equilibrates toward a pretty repeatable behavior. But interestingly, as we do so, the material actually changes its characteristic quite a bit. So this gray curve is the initial loading curve, and then this red curve is the loading curve after 10 cycles. We shifted it here. So of course, we have a permanent set. You see this here about 12%. So we shifted it over just for comparison purposes. And so this to us was also interesting because to some extent it explains differences that we see in data, right, that you just find in sort of these papers that I cited earlier, that preconditioning itself actually makes quite a bit of an effect. And then finally, we undergo the stress relaxation. And what we find is that, I mean, first of all, we have stress relaxation. I don't think that's surprising. And we did this actually in sort of 4 steps. And then we quantified actually the time constants that kind of determine this behavior. So we fit an exponential decay function with like several terms. And we found that basically if you include 2 terms, you capture this behavior extremely well. And interestingly, we find that the time constant 1 falls in the sort of range of a second, while the second time constant falls in the range of tenths of seconds. And this is interesting because although this is not proof, right, it kind of implies the existence of maybe 2 different physical phenomena that drive these viscoelastic effects. And so our theory was that one could be driven by the poroelasticity, so the water, basically the pressure gradient moving water and the water exchanging momentum with the surrounding matrix, and that sort of momentum loss being responsible for some of the energy dissipation, and maybe a second mechanism that's more diffusion driven. We don't know this at this point, but that's something that we are starting to study and understand, or at least try to understand. Okay, so this was the second step, but as I said, our ultimate motivation is to basically do fracture experiments and then analyze them in sort of an inverse finite element approach, in which we have material models that capture the mechanical behavior of this tissue outside of that fracture zone, right? So our plan is to use a cohesive zone element, but we need to capture all these other dissipative mechanisms in the surrounding material to be able to really identify what energy loss is due to the surface creation, what energy loss is due to damage, what energy loss is due to viscoelasticity, etc. So our plan was to basically capture all these behaviors that I just showed you in a comprehensive material model. And so this is kind of our third step. And so our approach was to say, okay, we kind of identified the hyperelastic material behavior and the viscoelastic material behavior. Let's formulate a material law in which we can capture that, right? Sort of a hyperviscoelastic material. And so this is a pretty standard approach. So we chose a viscoelastic material model that captures a large deformation far away from the equilibrium. This is a classic model by Rees and Govinci from 1998. So you see, this is not cutting edge research, right? We're talking about 25 years ago, but it's an outstanding material model. But then on top, we wanted to also include damage. And I'm not sure what the audience right now is, whether you're numerics people or non-numerics people. I try to keep the equations to a minimum. But one of the things we looked at here was to implement a non-local damage formulation as opposed to a local damage formulation. And essentially what this formulation does is, on a local level, it's sort of your classic damage approach in which you have some sort of damage function that has an evolution equation that sort of evolves a damage variable that reduces the energy stored. So in our case, of course, we have a hyperviscoelastic model. So we minimize the storage capability of both the equilibrium and non-equilibrium term. But that is what happens on a local level. And this is where a lot of the damage formulations stop, right? The issue with that is if you store damage only on a local level, then your damage fields ultimately become a function of your discretization. And so we essentially see non-convergent behavior in these damage formulations. And I'll show you this in a second. So we actually implemented a non-local damage formulation that was published by Waffenschmidt and many others. So this is not the only one, but this is particularly what we use. And essentially what we do here is we use a grading approach on the damage field, on the non-local damage field, to basically regularize the damage across our non-local volume. Anyways, long story short, this evolves our problem into a multi-field problem where we solve the damage field as well as sort of our displacement. So these are the governing equations for the nonlinear finite element method that we use to solve this problem. And because I'm at UT Austin, you really don't get around this. So we use the isogeometric framework to solve this. And so what we did here, we actually implemented a couple more experiments or different experiments for the purpose of informing this model. So we did an extension to failure. And because we didn't really find too much strain rate dependence in our viscoelastic experiments, we chose one strain rate. I think it's 0.1 millimeters per second. We did a cyclic tensile test, but instead of just repeating the same amplitudes, we sort of have an increasing amplitude here. So 10% we increase until it eventually fails. And then we did stress relaxation tests again, but we did them on individual samples. In retrospect, that was a mistake, but you're always smarter afterwards. And so the first thing we did was we just kind of looked at what does our model predict for a reaction force over displacement sort of test, right? So this is just a uniaxial extension under this geometry that you see here. And so this is a non-local damage approach, and this is a local damage approach. And the first thing we wanted to look at was sort of a convergence study of this. And so we underwent both H and P refinements, meaning that we just increased the mesh density, right? So that would be our coarse, fine, and finer. And we increased our interpolation scheme from linear to quadratic. And if you look at it, it's kind of hard to see, but I guess you can sort of trust me that this is our best approach, right? This is a quadratic approach with a fine mesh. And so you see everything kind of converges toward that. We can do this for the local approach as well. And I didn't blow this up here, but you actually see convergence as well. Okay, so this is kind of good news for the local approach, a little surprising to us. But one thing we found actually is these blue X's here is when the simulations ended, okay? So they kind of prematurely ended. We didn't investigate this too much because ultimately, we were going to go with a non-local approach. We just wanted to highlight its advantage. What does make it interesting, though, is when we look at the damage variable. So FD is sort of the multiplier that reduces the ability of our strain energy to be stored, so sort of to damage our material. And we see here convergence again of this curve as a function of H and P refinement. But if you look at the local approach, of course, we don't see that at all. And this is exactly what the problem is with the local approach, that we don't see convergence. It's kind of all over the place. And this is ultimately why we implemented this non-local approach. So this is not necessarily a brand new knowledge, but it's just interesting to see and directly compare for sort of a very practical study. All right, so the first thing we did was we took our data, so in red, for 3 cycles, right? So we sort of cycled 10%, 20%, and 30% strain. And then we inform our hyperviscoelastic model. And so we do an inverse approach, right? Basically an optimization problem where we identify the viscoelastic parameters. There are a total of 8 parameters. So 2 for the Ogden model and then 6 for the viscoelastic model with 2 viscoelastic branches. So basically 2 different time constants. And so this is the reaction force over time. It looks great. If we plot force over displacement, you can see a little bit of a discrepancy, but it's not too bad. So next, what we did was we kept those 8 parameters, and then we fit our model with damage to our uniaxial to failure data set. Okay, so this was the cyclic data set. This is the uniaxial to failure data set. So here you see the experiments. We load it, and then it just fails around here. If we use our viscoelastic parameter set without damage, we get this gray curve. And if we fit the damage to it, we see this black curve. Very important is, and you know, we are struggling a little bit with the reviewers here that say, oh, this fit isn't that good. It's very important to know that we're not fitting all of our data to this curve, right? What we're doing is we keep the viscoelastic parameters from our previous experiment and only inform the damage here. To sort of placate the reviewers, we did fit all of the parameters to this curve. And when you do that, you get a really good fit, right? Our model is able to capture this behavior if we identify it for this, just for this one data set. But that was not our goal, right? Our goal was to basically inform the viscoelastic model without damage. And the assumption here is that we don't have damage up to 30% uniaxial strain, and then inform the damage parameter independently. So basically to decouple that, that identification. And then to validate our model, we actually predicted unseen data. And specifically, we predicted the cyclic testing beyond the 30% that we used to inform the model, right? So basically, we use these 3 cyclical loading curves to inform our viscoelastic model. And then the rest is predicted. Okay, so this is unseen, and we didn't use this data to inform our model as a form of validation. Again, the force over time evolution looks really nice. We see discrepancies for sure in the reaction force. So you see here the simulations are in black, the experiments are in red. And here you see the damage evolution. And then finally, as a second form of validation, we predicted the stress relaxation tests completely unseen. So we didn't use this in the data information at all. And so we see actually pretty good fits for the 10%, the 20%, and the 30% strain, but then one data set just doesn't look right. And so I'm pretty sure that this is just an outlier in our experiments, right? These experiments are relatively sensitive to how we handle the samples, et cetera, because you can see that you have this sort of increasing reaction force, peak reaction force, which is what we saw in our original experiment. And then the last sample just doesn't really fit this pattern. And so our simulation, of course, doesn't know such a thing as, mishandling of the sample, et cetera. So we don't capture that. Again, if we fit these individually to just this data, so we don't use it as a form of validation, but instead just do curve fitting, we see almost perfect fits, right? So it's not so much a matter of can we fit these data, it's more of a matter of validation, right? Can we predict unseen data? And I think, to me, in my lab, I try to communicate this with my students, that there's a huge difference between having good fits and being able to predict the behavior. And so whenever I review papers, I urge the authors to show me that their model can actually predict unseen data, and then it never looks as pretty, right? And so that's something that I'm struggling now and defending sort of our approach against reviewers. And I'd love to hear your opinion on that. So this is the work that we've done. So these are 2 papers that we've published, as well as 1 paper that we submitted that we're struggling a bit with. But what we're doing next now is to basically do these other parts of my vision for the mechanics of blood clots. We want to look at fracture as well as at the micro scale, right? And so one of the things we started doing is actually we take the blood clot. And what's very cool is you can take the blood, and it consists essentially of 3 things: the fibrin, the platelets, and the red blood cells. And we can actually put them in a centrifuge, spin them down, separate the 3, and then we can fluorescently label the fibrinogen or the fibrin. and fluorescently label the red blood cells and the platelets, we can put them back together and make blood clot out of it. And then we can look at it, in this case, under a 2-photon microscope. And so what you see in green is actually the fibrin mesh, and what you see in red is the red blood cells. So now you can imagine we have this blood clot fluorescently labeled. We can actually do now mechanics on this. And so we actually started doing this with my collaborator, where we basically build a little uniaxial tensile testing machine and a little shear tester under a 2-photon microscope, and we can look at the fibrin mesh under deformation. And one of the things we can do is, for example, we can look at the probability density of fibers to have a certain in-plane angle or through thickness angle. And so then we can kind of build a statistical model that we can incorporate into our macro models as a multi-scale approach toward understanding how the micro scale gives rise to the macro behavior. And then also, we've started actually doing these fracture experiments now that we have a material model to model the blood clot material. So this is your classic, mode 1 fracture experiment. And so this is a video that we recorded. So you have your initial notch, about 1/3 through thickness, and then we displace this at a given rate. And you can see here the crack propagate, right? So then the idea is that we build a basically finite element model and then look at sort of the critical fracture energy that is required to begin propagating this crack. Yeah, this is the overview that I wanted to give you and hopefully delivered. I very much want to appreciate the support that this lab has received from the American Heart Association and the National Science Foundation. And if any funding agencies accidentally watch him, I'll be happy to receive more funding from them. And then once again, also thank you for the invitation to give you this overview, as well as to my collaborators and students who've done an outstanding job helping me with this.